Hello, everyone. Thank you for being here in the last section from the last day. Today, I present you part of my PhD that is working with some fast divergency species, try to understand the main drivers of their divergency. I'm staying here because I'm using a PDF and I don't have a pointer. So I just will be here to point in things. Yep. So you hear me? Yes. So I work with Petunia. I'm sure that everyone here haven't seen Petunia around, but the one that you're seeing around is the Petunia hybrida. I'm telling you today about the wild Petunia that is endemic to South America, and their divergency was very influenced by the Pleistocene, and they divergence very fast and very recent in about 16 to 20 species. Species, taxa, lineages. This is hard for them because they fast diverge. You're going to see how hard it is to delimit them. This is one of the oldest phylogenies we have from the genus. We, in the lab, are trying to update this phylogeny by the end of my PhD. But the lab has been working with this population and species for the past 20 years, so we have a very idea of how they arrived and how they evolved in this location. First, we are in the south of South America, from especially their origin in the lowlands of Brazil. And then they expanded during the Pleistocene, and they reach all the other parts of their area of distribution, and arrived in the highlands. We have a highland in Brazil. It's not that high, but it's high enough. In that part that is marked in the map, and the highland species are this one highlighted in the phylogeny. So in this work, I was trying to understand what was the main drivers of their diversification. And to this, I chose 4 of the highland species and 1 lowland species, interior. Interior has the widest distribution of Petunia in the lowlands, and we believe that they have a very important role in the colonization of the highlands. For this, we collected samples for the populations of all these species across all their distribution, and the populations of P. interior near the highland species. One important thing to say is that their environment is a patch-like, island-like environment of patches of grasslands surrounded by Araucária forest or even Atlantic forest. But this fragmentation was very recent. In the end of the Pleistocene, the Araucária forest expanded and fragmented these populations or species. For trying to understand their drivers and what their relation between these species, we used a whole genome sequencing, a DART-seq, similar to a RAT-seq. And with 8,000 SNPs, I've done a bunch of analysis to try to understand their relationship. And here I'm going to show you some of these results. First, they have, for now on, I will say lineages, because I think it's the best way to describe them. There are some lineages that are very structured. You can see by the reddest colors. And we saw that the geographic distance, the geography of these populations, or the lineages, were very important for their structuration. I did this analysis, the construct analysis, that is tested models using test the geographic distance, the structuration with geographic information or without the geographic information, and the best model was using geographic information. You can see here the sites are very structured. And I searched of how much of this information, this geographic information, was important to the variation. And I found that 53% of the variation, the FST, could be explained by geographic distance and environmental variables. And here you can see like 12% only geographic distance, 18% the environment, and both together because they are always related. Explain the structuration. So in these populations, in these lineages, I think one of the most main drivers of diversification is isolation by distance and also isolation by environment. But not only this, there are more things happen here. Here we can see the PCA and some of structural analysis that there are a lot of going on here, and I'm going to dive in some of specialties within these results. First, this here was supposed to be 2 species. But in all structural analysis, they are closer together. And only when analyzed alone, it was possible to differentiate them. You know, okay, what's happening? I already knew that they live in a little different habitats, so I tested the niches of these populations, and this analysis shows in a similar way to a PCA, but with the environmental variables. And you can see that these 2 lineages has very different niches. So with this, I'm thinking that probably one of the main drivers for these 2 lineages was ecological adaptation, that probably some genes was important to this differentiation, but the whole genome stay similar. For these 2, this was a little more complicated. I did a whole other paper just trying to delimit these 2 species. Long story short, there were supposed to be 2 species, and then a taxonomist are saying, no, this is 1 species. And genetically, there are 3 lineages. And in this other paper that I present the poster on the first session, I have the conclusion that could be 2 species, but this one species, and this as another species, but we can see there they have a structuration between them. And in this paper, I could find that this is a snack network, and we can see there's some hybridization or integration within their evolution. But I think was one of the most fun things that I found when analyzing these lineages was this one. When I say first that pink pay interior was one of the lowlands species that we believe that have an important role colonizing the highlands, here they divide in 2 groups. They're supposed to be 1 species, but divided in 2. And here in the structure part, in the APC, they're a completely different cluster. And in the structure was a mix between 2 different genetic components. And I was like, what they are these? And it's 3 individuals, only 3, from the same collection site. What they are? They are hybrids. What's happening in them? And I can see that they share a genetic component with the highland populations. And here, they are closer to the highlands than their original population. So, okay, maybe this one is hybrid between some, or there's some integration between the other ones, and that's why they're closer here. I tested this, and I tested in this ancestral painting and other analysis, but this one I think was the most fun to see, that they could be hybrids among all the other species. And I was, how this is possible? And thinking a little bit more in this, I realized that maybe it was not hybridization. And yet, that these three individuals holds ancestral polymorphism. In all other works in Petunia, we always say, oh, they share a lot of ancestral polymorphism, they have a lot of divergency together. And I think I found some of the remains of the ancestral population that colonized the highlands in these three individuals. As you can see, they share a lot of variation with all the other populations. And, as I say, in this ancestral, what I think it is, these three individuals are remains of the ancestral population that colonized the highlands. And they had the variation that was important to after the diversification that happened when the Pleistocene arrived and the environment began to change the weather. Like here. Here is the population, the collection site of these three individuals. So I believe they just expanded their diversification, they expand to the highlands and arrive in other parts of the highlands, and then started the speciation and diversification in these sites. And I can see evidence of this in other analyses. First, this one I can see they are closer to the others, but also in the administrative tools tree, they are in the base of the highlands. In this tree as well, they are here most closer to the ancestral of the highlands, and also in the snap tree, there are different branches. Like, believe in me, there are some other branches here. They have different histories trying to tell the phylogeny of this one. So this one thing that I believe that I found a source of variation to their diversification. But also in that snack network, this was weird. Now I don't have an answer exactly what happened here, because I'm saying that the highland one has some interaction with the ancestral population here. But overall, hybridization is a thing that happens with Petunia. So there's some gene flow or integration from this population to this one. And with this, just showing you how complex species divergence can happen, and when they are experience climate change, and how fast this can happen, because everything that I'm telling you was during the last 2 million years. So we have example, isolation by distance, ecological adaptations, hybridization, and integration. For the next steps, I need to resolve this beautiful phylogeny here that I'm trying not to be crazy about. But if anyone have any tips on how I can work with this, I'm glad to have any tips. And that's it. Thank you. I'm finishing my PhD this year, so I'm looking for a postdoc position for the next year. Thank you.
All right. Well, thanks everyone for coming. My name is Brian Waldron. I'm a postdoc right now at Virginia Tech, but I recently finished my PhD at Ohio University last year, studying species formation and species delimitation in plethodon salamanders, so woodland salamanders. So there's an emerging consensus that species are really evolutionary lineages. So rather than being defined by reproductive isolation or morphological distinctiveness, really the essence of what a species is are these ecological or evolutionary lineages. And more precisely, they are segments of separately evolving metapopulation lineages. But of course, species in practice are a lot harder to diagnose. The right part of the figure shows the reality for many species complexes, in which we not only have divergence, but we also have gene flow, full-on lineage fusion, the expansion and contraction of populations, local extinctions, all ongoing while we have these incipient lineages forming. among the groups. So when we look at this figure on the right at time t1, it can be really challenging to say, is this part of one big inclusive metapopulation, or are these multiple component lineages, are they worthy of being recognized as separate species? So when we think about this challenge, a lot of times we might think about these rapidly evolving lineages sort of at these very shallow timescales. Maybe we think about these adaptive radiations into Knowles or Darwin's finches, in which morphology might be evolving at different rates than the ecology. You might have geographic isolation among some, but not other groups. And maybe the genomics don't show a signature of genealogical divergence just because of how rapid these species are. So they're often thought to be this worst case scenario for species delimitation, where really we, depending on what lines of evidence we use, we might come to different conclusions on the number of species in our data set. But I want to focus on a different kind of problem, what we call non-ecological species formation. And I call it a different kind of worst case scenario, because these are woodland salamanders, so these are 10 species within the woodland salamanders of very, very morphologically similar species. Some of them are actually identical, or at least identical based on characters we've been able to study. And so what this does is it puts us pretty much at the mercy of genomic data, or in the case of these, allozyme data was used to describe many of these species in the past. And that might just be all we have available to describe species. And we're going to zoom in on one species here, the eastern redback salamander. And the reason we're going to do that is because the eastern redback salamander, Plethodon cinereus, it has the largest distribution, not only among its close relatives, not only in the genus, but of the lungless salamanders as a whole. It has the widest distribution. And this is really not trivial because lungless salamanders make up 63% of salamander diversity. So this gray distribution that you can see is as big as it gets for the family. And this is really striking because lungless salamanders, but especially these terrestrial woodland salamanders, tend to have dispersal distances only on the order of meters. They can move further in rare cases, but they tend to be very geographically, they tend to evolve genetic differentiation very easily on small scales. In some cases, hundreds of meters, you can detect isolation by distance. So vast genetic structure. So this really leads naturally to this question of like, is this big distribution truly representative of one single species? Or could we have genetic lineages that are worthy of recognition? So we had some work with mitochondrial DNA that came before me, in which these 6 mitochondrial clades had been recognized throughout the distribution. I don't know if my pointer can see. But you can see these, they vary quite a bit in range size. So you can see some narrowly distributed groups in the southern Appalachian Mountains, as well as this northern clade, these blue triangles, which wrap around the eastern, northern, and western periphery of the distribution. So these were these testable hypotheses for candidate species in the range. So what we did is we took a subset of these samples and we sequenced them using anchored hybrid enrichment to get about 300 loci throughout this distribution. And our sampling was really targeting kind of the core areas of each of these clades, as well as areas of contact, to see if we could pick up any evidence of admixture, gene flow at these areas of contact with our nuclear data. So jumping right into population genomic structure, you can see these 3 groups in the southern area of the distribution, somewhat corresponding with mitochondrial groupings. You can see groups 1, 2, and 3 in the southern area that kind of occupy these somewhat more range-limited, albeit widespread compared to other species, distributions. And then in the north, we have these slightly wider ranging groups 4 and 5. So we will zoom in on the core area of the distribution here. And one thing that's really striking is that even with really, really coarse sampling, at every area of contact between these groups, we see evidence of admixture. So between groups 1 and 2 in the very south, you can see admixture. Between groups 4 and 5 in the north, you see what's potentially a pretty extensive area of admixture going along the valley and ridge. If you look all the way on the western portion of the distribution, there's an isolated member of group 2 in northern Kentucky, and even there you see admixture with the northern populations. And we also see in the central area of the distribution, there is, I don't know if I have any point here. There's a 4 population contact zone. So this area of extreme diversity where groups 2, 3, 4, and 5 are all in contact exhibiting admixture, and one sample even has the genomic ancestry of 3 groups. So it seems that these populations are really interbreeding at any place that they have the opportunity to do so. When we look at this in a phylogenetic context, we see still evidence for these 5 groups, and all of these admixed samples tend to be early branching from the core genomic group. This is a pattern we see a lot, so that hybridization kind of creates these early branching pectinate lineages. And when we remove those samples, we again see support for these 5 genetic groups that kind of have this successful hierarchical level of divergences. So we also wanted to do this analysis while accounting for gene flow. So we use the program TreeMix to account for historical gene flow, and we call this historical gene flow because we've removed the individuals that are at contact zones. So we're kind of removing what we think is contemporary evidence of admixture and focusing on ancient cases of gene flow. And again, we see lots of evidence for reticulations throughout this tree between many pairs of groups. So this is evidence that we not only have connectivity today, but we've also had connectivity between these groups historically. Now, finally, we wanted to look at this in a species delimitation framework to just understand what kind of evidence we have for multiple species. So when we use the program BP and P, we always got evidence for having 5 species in the data set, which is not super surprising because BP and P tends to recognize population structure as independent lineages. But one thing that is somewhat striking is that this analysis is showing even included our admixed samples at contact zones, so that didn't seem to reduce support for having multiple species according to BP and P. So we wanted to take a somewhat more conservative approach to species delimitation here. So for this, we use the genealogical divergence index, so it uses those parameters from BP and P in terms of divergence times, population sizes, to assess evidence for lineage independence. So the GDI is basically a function of divergence times and population sizes. Longer divergence or smaller population size, more evidence for lineage independence. So the value ranges from 0 to 1. Rules of thumb kind of thresholds are that values above 0.7, evidence for distinct lineage, values below 0.2, lack of evidence, and values between 0.2 and 0.7 are this kind of gray zone in which the evidence is ambiguous. And so when we run this analysis and we include all samples, including these admixed samples in the data set, you can see our outgroup sample, the southern redback salamander, falls out by 1, which is good, sanity check. But the remaining groups tend to have very poor support for being multiple species. The highest value we see is around 0.5 in our southern group, in which we have like a pretty small, narrowly distributed sample for group 1. So when we remove admixed samples, because the jury is kind of out on like how we want to treat these contact zones, should we include them in the analysis or should we exclude them? When we exclude them, we do see increasing support for having multiple independent lineages. So all of these samples go into all of our groups, seem to have evidence in this gray zone of species formation. But group 1 seems to have evidence of being an independent lineage. So basically we see increasing support, most of these in the gray zone, but we do have one group that seems to have evidence of a distinct lineage. However, that was one group in which we only had a single locality available. So one thing we wanted to do is assess what is the consequence of using a single locality to represent an entire genetic group, which matches what we have for group 1 in terms of sampling. It's a very narrowly distributed group. But at the same time, it might actually represent something real about we might have a montane species that recently underwent a range contraction and is only found on a single mountaintop. Or we might just have cases where species is hard to find, and that's what scientists have available to assess independence for species. So when we subsample every group down to one locality, we see a greater inflation of this GDI. So basically all of these samples are pushed towards the line of evidence of having an independent lineages, and including 2 groups now that have support as being independent species if we took this rule of thumb threshold. So it seems that subsampling or using a small number of samples is inflating evidence of lineage independence. And this is probably because we have a lot of population substructure within these groups, and all of that is lost when you only use a single sample from a single locality. So to summarize, Plethodon cinereus, it seems to be a really good example of this gray zone of species formation. So we have these 5 groups that are very clearly defined and phylogeographically structured, but according to GDI and our other analyses, we don't seem to have good evidence that these should be recognized as independent species because we have contemporary contact zones, historical contact zones that must have been maintaining some connectivity between groups, and the GDI, at least when we use admixed samples being excluded or including all samples, it doesn't really give us strong genomic evidence that we have multiple species. And we conclude that Plethodon cinereus really is best representative of a single metapopulation, kind of pushing the bounds of how inclusive we should be in our definition of what a metapopulation is before we might want to start describing species. So we have these 5 phylogroups that have really been connected by contact zones and these pulses historically of gene flow. But this really might be illustrative of how these cryptic species complexes form more broadly, where you just have continuous production of these incipient lineages on the landscape, and only over long periods of time do you really get strong evidence of having independent species. And finally, these large structured metapopulations also present challenges for species delimitation. In an ideal world, I would be able to model the demographic history for all 5 groups or some of the substructure within those groups, getting at independent population sizes, changes in population size through time, different rates of gene flow, but the model space becomes huge for these kinds of problems. So getting better methods that can better explore that model space and come up with realistic demographic models for these vastly structured species is really going to help us in the future with similar groups. So this work was recently accepted at Systematic Biology, so hopefully you will be able to read more soon. So yeah, I just want to thank all my co-authors on this work, and like I said, I'm now at Virginia Tech working with Josef Ueda, so you can contact me there at any time, and if I have some time for questions, I'd be happy to take them.
Hi, everyone. Thanks for making it to the final session. My name is Eamon Corbett. I'm a PhD student with Brant Faircloth and Rob Brumfield at LSU. And today I'm going to be talking about boat-tailed and gray-tailed grackles, phylogeography, hybridization, and eye color variation. So to introduce you to these species, if you're not familiar with them, these are large social blackbirds in the family Icteridae. They're closely related, and they form a species complex. The boat-tailed grackle is essentially a marsh species. It's found in the US from Connecticut to Texas, and it has 4 subspecies that vary in eye color from yellow to brown. And then the great-tailed grackle is a much more widespread species. It's expanding rapidly, often found in human habitats. If you've ever been in a gas station parking lot in Texas, you've probably seen great-tailed grackle. And it has 8 subspecies, but always has light yellow eyes. And actually, there's a third player in this species complex, the slender-billed grackle that was endemic to marshes in central Mexico, but unfortunately went extinct around 1910, likely due to the draining of those marshes and loss of habitat. So why am I interested in studying grackles and studying this system? 2 main reasons. One is they're a potential model for studying speciation and hybridization, and then the other is because of that eye color variation that exists within the complex. So I'm going to go through a little background on each of these points. First off, these 2 species were originally conspecific, but they were also allopatric prior to the 20th century. That dramatic range expansion in great-tailed grackle throughout the 1900s has brought the 2 species into contact in Texas and Louisiana near the coast, where they're now broadly sympatric. They were split based on evidence of assortative mating in that contact zone, but there is some evidence of possible phenotypic hybrids, at least. And one actually additional little twist is that based on mitochondrial DNA, it seems like gray-tailed grackle may actually be a paraphyletic species with an eastern clade sister to boat-tailed grackle and a western clade sister to the extinct slender-billed grackle. And then zooming in on the eye color variation just in boat-tailed grackle, which is the variable species, we have a light-eyed species on the Atlantic coast, a dark-eyed subspecies on the Atlantic coast, a dark-eyed subspecies in Florida, a light-eyed subspecies in Alabama, and a dark-eyed subspecies in Texas and Louisiana. So we get this sort of leapfrog pattern of eye color distribution along the coast. But actually, I should point out that it's maybe a little bit more complicated than that. These are a bunch of photos that I have taken all in Louisiana. These are supposedly the dark-eyed subspecies. So as you can see, there's actually quite a bit of variation. This is at a couple of localities very close to each other. And then I mentioned that these are gas station birds. This is a gas station in Moss Point, Mississippi. And you can see that with the zoomed-in insets that we have a light-eyed bird and a dark-eyed bird at the same pump. So, so it's probably better to consider these two sort of northern Gulf Coast subspecies as being variable-eyed, with one of them primarily light-eyed, one of them primarily dark-eyed, but with a lot of variation. So my research questions are, first off, looking at is great-tailed grackle parapatric, as has been suggested with the mitochondrial DNA. Looking at the eye color variation in boat-tailed grackle, whether the genetic structure that we see corresponds to that variation or more to geography. And then also, do we see any evidence of hybridization between the two species where they come into contact? So to do that, we sampled 115 individuals from every named taxa, so all 13 subspecies in this complex, 63 boat-tailed grackles, 51 great-tailed grackles, and we had one topad from 1910 of a slender-billed grackle. And we did targeted capture of UCEs. And most of these were from museum specimens, but there were also some, in particular those Alabama birds. There were no tissues in any museums anywhere, so we had to go out into the marsh to get them, or diving into the elephant ear or the Atchafalaya River to get some samples. We ended up collecting 20 samples of that subspecies that are now at the LSU Museum of Natural Science for anyone to use in the future. So this was our sampling across the complex from New York south to Peru of the two species, and you can see that extinct slender-billed grackle near Mexico City. And then we built a RAXML tree, and I should mention that the last batch of this data we just got off the sequencer like a week ago, so this is a somewhat preliminary RAXML tree, but I think the main points will still stand. We see a big clade of boat-tailed grackles at the top, a big clade of great-tailed grackles, and then the one slender-billed grackle comes out as sister to the great-tailed grackles. So you'll notice that, are we seeing a paraphyletic great-tailed grackle? Not really. In this case, the great-tailed grackle is coming out as a single monophyletic clade sister to slender-billed grackle. So that suggests that there's some disconnect between the mitochondrial data that had been previously published and what we're seeing in the nuclear data. I said this is fairly new information, so we're going to do a lot more investigating to see where that disconnect may be happening. Then if we zoom in at just boat-tailed grackle, we get sort of two main clades. There's a clade that includes the Florida and Atlantic coast birds, so the dark-eyed and light-eyed birds from Florida and the Atlantic coast, and a clade that includes all of those northern Gulf Coast birds, the Alabama and Louisiana populations, as well as Texas. And neither of those are reciprocally monophyletic, but the two main clades are. So if you plot that on a map, this is where the division between the subspecies is. And that actually does correspond to a small gap in the range. It's the only gap in its range in the Florida Panhandle where there doesn't seem to be sufficient salt marsh habitat. But the interesting thing is that you have light-eyed birds and dark-eyed birds on one side of the gap and light-eyed birds and dark-eyed birds on the other side of the gap. So the relationships between subspecies seem to be corresponding to geography and not to eye color. And that's actually going to be helpful for us in our next stage of research, where we're hoping to look at the genetics of eye color variation in this species, because we have sort of replicate populations that are not very genetically variable, but they are very phenotypically variable. So we have a SNPs only called for a subset of our samples, 42 samples, that include great-tailed grackles from the contact zone and from allopatry, and boat-tailed grackles also from the contact zone and from allopatry. So we constructed a structure plot to see how these are related. And this is what it looks like with all of our boat-tailed grackles and our sampling of great-tailed grackles. And so the first thing that should jump out is there's a pretty strong boundary there between the two described species. And that actually becomes even more striking when you look at, this is the contact zone, so all the birds in this box are in the exact same locations in coastal Texas and southwest Louisiana, and yet we seem to be getting this really pretty strong barrier, no real evidence of intergression between the two. So do we see evidence for hybridization between these two taxa where they're sympatric? No, we haven't found any evidence of that. As reported by Sealander and Giller in the 60s, there seem to be some barriers to reproduction between these two species, whether that's behavior, they do differ in song. It is fairly surprising, though, because they're quite similar. They can be difficult to identify in the field. They're difficult to identify as museum specimens. So it's interesting that they seem to be fairly good at identifying each other. Or there's a barrier, a post-mating barrier that's keeping them apart alternatively. This doesn't mean necessarily that there are never any hybrids. There could be occasional F1s, but it does suggest that there's not widespread gene flow because we're not seeing any evidence of genes crossing that boundary between species. So looking forward, where we're going next with this research, we've sequenced a reference genome of the boat-tailed grackle, a PacBio reference genome that should be coming out pretty soon. And then we've done resequencing to about 10x coverage of 55 individuals of known eye color. So mostly from, and we're going to be looking for loci associated with that eye color variation. Most of these samples, and all the ones pictured here, are from those variable populations in Louisiana, Mississippi, and Alabama. It's actually the top two rows are Louisiana, the next row is Mississippi, and the bottom row is Alabama. So there's maybe something of a cline if you squint at it, but there's also a lot of variation at single localities. So we're hoping to be able to leverage that to try to look at the genetic underpinnings of this variable trait. And then also we'll be able to compare that. We have samples from the non-variable, entirely dark-eyed Florida populations and the entirely light-eyed Atlantic coast populations. So we'll be able to sort of validate anything we find in the variable population. So just to sort of summarize, boat-tailed and gray-tailed grackles appear to be coexisting without extensive integration, despite their phenotypic similarity. And tentatively using UCEs, gray-tailed grackles appear to be monophyletic, which may conflict with the mitochondrial data, so there may be something interesting going on there. Bow-tailed grackle populations are grouping by geography and not by eye color, so we're getting a lot of eye color variation that doesn't correspond to neutral genetic variation. And so for these reasons, I think grackles are going to be a great system for studying eye color variation and also the dynamics of speciation. So there's a lot of people to thank, particularly a lot of people at museums to thank for lending specimens and for facilitating this research, and hopefully also given back to museums by depositing all of our samples as well. And I should have some time to take any questions.
Okay, my name is Tobias Van Elst. I'm a PhD student at the University of Hannover. And I really appreciate that so many people made it to the last session. So I'm going to talk about phylogeography of microcephalus species in Madagascar. So let's start with a quick overview of Madagascar. The island is separated for about 80 to 90 million years from all other continents, and it is really characterized by a complex physical geography. So on the east coast, we have these. Maybe I'll do it like this as well. So on the east coast, we have this escarpment in elevation towards the central highland plateau with elevations above 1,000 meters. And then on the west coast, this elevational gradient is much less pronounced. And this really results into a diversity of habitats as humidity from the Indian Ocean precipitates here into on the east coast, forming this band of humid rainforest. And then the western parts and the southern parts of the island in particular are much drier with dry deciduous forest and arid shrubland. Madagascar is also well known for its high levels of biodiversity, with much of it is endemic. About 90% of vertebrates are endemic and about 80% of plant species. And new species are continuously being described even today and even in primates. Madagascar's biodiversity entails the iconic lemurs, and part of this, about one-fifth to one-fourth of all lemur species are mouse lemurs. So this genus comprises the smallest of all primates. They're about 100 grams in weight. They are arboreal nocturnal. About 25%, 25 species, although the species number is highly controversial. We just got a paper accepted that suggests to synonymize actually 6 of these species. And 80% of these 25 historic species are threatened by extinction. So due to this high biodiversity and the complex geography, Madagascar has often been proposed to be an ideal laboratory to study diversification processes, and particularly lemurs due to their, or most lemurs due to their species diversity and relative ease of capture compared to other lemur species, for example, are a suited model. So what hypotheses have been proposed to explain biodiversity patterns in Madagascar? So most models are allopatric speciation models promoted by an interaction of paleoclimatic fluctuations and the island's geography. So for instance, we could have a refuge model where a formerly connected population is separated into these distinct refugia during cooler and drier conditions, such as the last glacial maximum, and then we have divergence in speciation in allopatry. Or a simple riverine barrier model where a formerly connected population is separated by rivers. And also watersheds have been employed to explain connectivity and isolation patterns or cycles in Malagasy taxa, as populations might be able to maintain connectivity in favorable conditions across these watershed regions that enable crossing riverine barriers. And then when the climate becomes drier, these watershed corridors might disappear, and this is particularly the case at lower altitudes where arid conditions are much more pronounced during glacials, and then we could have divergence in these distinct refugia again. So the aim of this work was to investigate how these models can explain the divergence of Microcebus or mouse lemur species in Madagascar's humid forests. And for that, we conducted a comparative assessment of population structure, diversity, and the determinants of this structure in co-distributed Microcebus species in a center of biodiversity in northeastern Madagascar. So this is our study region. And you can see again that we have this elevational gradient from the east coast towards the central highlands. And then the entire region is separated into 17 inter-river systems, which are indicated by the numbers here, by rivers of different size and different watershed altitudes. And so we sampled this region extensively, trying to fill all the inter-river systems at lowland sites and at highland sites to really detect these migration patterns around watershed regions. And there are 4 Microcebus species in this region. And in this talk, I would like to focus on these 2 just because of the limited time. So we used red-seq sequences as markers, and then we inferred phylogeny, population structure, diversity, patterns of gene flow. And then we also fitted isolation by resistance models using elevation, topographic heterogeneity, flow accumulation as a continuous proxy for rivers, climatic niche models, and forest cover as predictors for genetic diversity or connectivity. So, and here you, for example, you see the climatic niche suitability of this one species, Microcebus jonahi, in the region. So let's start with Microcebus jonahi. On the left-hand side, you see the admixture proportions in the pie charts, where the pie size is proportional to sample size. The arrows indicate rates of gene flow inferred under a coalescent model, and then the crosses are absence records. And what we can see here is that we, and please note that we included its sister species, Microcebus mercaturi, here, because the species status was not very clear beforehand. But the results are congruent if we exclude this species. So what we can see is that we have these, at best K, we have these 6 differentiated clusters with patterns or rates of gene flow and admixture predominantly between inter-river system 6 and 11. And there in inter-river system 9, you have the highest rates of admixture. And this is also evident in the principal component analyses, where we have this gradient-like structure of genetic distances of these populations. And then to the far south, the populations in inter-river systems 12, 13, and 14 are much more isolated. And in the principal component analysis, I did not include Microcebus mercaturi because this would just shift all the Microcebus jonahi populations into a single cloud, and you wouldn't be able to see anything. So the next step was to understand what determines this population structure. So we first visualized deviations from a simple model of isolation by distance, which is the map on the left side. So red areas indicate higher genetic distances than what we would expect from an IBD model. You can see that there's a lot of red there, and it seems to be at least overlapping to some extent with these rivers here and here and here. But we investigated this in more detail with our isolation by resistance models and found that the best fitting model included flow accumulation, climatic niche suitability, and then topographic heterogeneity as predictors. And this is what you see in the diagram. So the x-axis is the scaled variable value, and the y-axis is the conductance, which is the proxy for gene flow. And when we start with flow accumulation, we can see that given a specific value, the gene flow tends to go against 0. So rivers really seem to be a very stark barrier, which we would have expected. And then climatic niche suitability is positively correlated with gene flow. And we kind of expected that we would find elevation in this model, because if rivers are a barrier, then the only way to cross or go against these rivers would be across the watershed regions. And given that the species has such high structure, this should be difficult for the species. But instead we find the climatic niche suitability, and this is only moderately negatively correlated with elevation. So this indicates that elevation alone cannot explain this, but there seem to be more intricate climatic factors playing a role in limiting the species dispersal. And then interestingly, we found this topographic heterogeneity in promoting conductance, and our current hypothesis is that these regions that are more topographically complex might provide ecological buffering effects against interannual or seasonal climatic variations, because maybe they are more prone to retaining humidity and providing more stable environmental conditions. So then when we looked at the phylogeny, these population structure findings were more or less confirmed. And given that the sister species is distributed in the far north of this species, and that we find this decline in genetic diversity here for Microcebus jennai in red, with lower genetic diversities in the south of the region, we hypothesized that the range expansion of this species started from a smaller distribution into the towards the south, potentially from refugium, because the diversification of this genus happened within the last 2 million years. So patterns we find in Microcebus laetzara are quite different. So here we have k equals 2 as the best number of clusters. We have this gradient index mixture when we go towards the north. So mixture proportions increase when we go towards the north and around this Antainambalana, which is the large river here, over these highland regions in the far north, and then towards the inter-river systems 2, 3, and 4. And this gradient-like structure is also indicated in the PCA, which shows a much more gradient-like pattern. This is also congruent, and then when we look at these gene flow rates in more detail, we find that gene flow is predominantly directed from the highlands into the south, into the lowland regions. And this is congruent with genetic diversity being much higher in this species. So this is the green plot here. And all other species in red, blue, and yellow have much lower genetic diversities. And notably, this species also occurs south of our study region, up to 400 kilometers. And there we have in black similarly high genetic diversity. So this connectivity really seems to be homogeneous across the entire range of this species. And then similarly, we looked at deviations from an IBD pattern, and there we have a much more open migration surface with these negative deviations from an IBD pattern mostly coinciding or mostly being here in the north. And this area here in the bay is likely caused by the high genetic distances between inter-river systems 3 and 5, which coincide with this large Antainambalana river again. And when we look at the isolation by resistance model, we again find flow accumulation. So rivers are a similar barrier for this species, which we would expect because mouse lemurs are really bad at swimming. And then we have forests, which we would also expect since it's an arboreal species. And then we also have this topographic heterogeneity in this species. And interestingly, we do not find climatic niche for this species, so which supports the hypothesis that this species is much more flexible in traveling around these higher altitude regions. When we look at the phylogeny of Microcebus laelatsara, we have at the bottom we have all the southern populations that are not shown in this plot, and then we have this successive splitting pattern from southern interriver system towards northern interriver systems, which suggests that the species successively colonized the region from its more southern distribution. And just as a comparison to Microcebus jonai, we have similarly high genetic diversity across the entire range. So in conclusion, we find significant differences in connectivity and diversity between these species, and these are likely caused by different responses to different landscape features, although some landscape features seem to have a general role in all Mouse species, like rivers and the topographic heterogeneity, but then climatic niche suitability and potentially elevation seems to play a different role for different species. We also identified the highland regions as important dispersal corridors for both species, although Microcebus jonai is more restricted in these, which has conservation implications to facilitate gene flow between these isolated populations, and potentially these topographically complex regions could also be targeted, but we need to look into that in more detail. And then we built an informed hypothesis regarding the diversification history of the two species, but in the next step we will have to test these really with more detailed demographic models. Yeah, and with that I want to thank my co-authors and all the Malagasy people in particular that helped and made the fieldwork possible, and our funding agencies. Thank you.
Thank you for being here. My name is Rozenn, and I'm currently a postdoc at the University of Chicago, working with Julia Crainier. But right now I'm going to present you the work that I did during my PhD with Will Radcliffe on the very right and Zach Gompert, and also in collaboration with Karen Mock and Aurore Pajot. And this work was partially funded by an SSC grant, the Rosemary Grant Advanced Award. I would like to start this talk by talking about the importance of mutations, which are the raw material for evolution. So mutations simply are a change in the sequence of an organism's DNA, because every time a cell replicates its DNA, it's prone to errors, like here simply you have a C that changes to a T. And this is important because sometimes these changes can alter the phenotype. So for example, here if you look at this picture of this tree and focusing on this branch, right here you see that it's much more foliated than the rest of the tree. And this is because there was a single mutation that occurred at the beginning of the branch growth that rendered the whole branch resistant to defoliation by beetles. This is called genetic mosaicism, when you have some mutations that are present in only some parts of the organism and not other parts of the organism. And usually most of us are thinking about sexually reproducing organisms, and these somatic mutations don't really matter for the progeny, because only the germline mutations are going to matter, so this bottleneck allows these somatic mutations not to be passed down. But when you focus on clonally reproducing organisms, this mutation that appears in one of the modules can be passed down to the next modules. Furthermore, these clonally reproducing species are important because they represent 35% of the animal phyla and 65% of the plant phyla. And we also know that they can achieve exceedingly long lifespan, which is yet another opportunity for mutations to accumulate. So what can we learn about an organism's evolutionary history based on the spread of somatic mutations? So to look at this question, I focused on one particular organism that is a giant and still living woody organism, the pendoclone. It's located in Utah in the Fish Lake National Forest. The pendoclone is a quaking aspen, Populus tremeloides. It's about 43 hectares and made of 47,000 stems that are all connected by the roots because they all arouse from a single mother tree. And as you can see on this picture, so the green dots are the pendoclone samples, and it's surrounded by approximately 61 other different clones. And currently, we do not know anything about its spatial structure or its age. So what we think happens is when a seed germinates, it gives rise to a tree, and from the roots, new ramets can grow. What's interesting about these clonally reproducing trees is that each ramet can only live up to 150 years max. However, the root system stays underground and can survive longer, so that the whole organism can be older than its parts. So what we were thinking is to follow mutations. For example, if a mutation appears in a root, we could technically find it in the progeny of this root. So the questions that were of interest to me in this project was to characterize the pendo structure in space and in time. And I'm also interested in looking at the mutation structure within and between units of the clonal organism. But for the sake of this talk, I'm only going to look at these two first questions. So to look at these questions, I went in the field and I sampled at two different scales. So this is the large-scale data set where we sampled leaves from the pendoclone and the surrounding clones. And I also looked at the finer scale data sets. So you can see here, this is like a zoom in, and we had two subparts of the pendoclone where we sampled leaves, roots, and bark. And we also have a third data set that is 12 samples that we sequenced 8 times. So the same DNA extraction that we just technically sequenced again. So the first thing when I got all of the data was to identify which samples pertain to the pendo as compared to the neighboring clones. So you can see on this ordination plot that there are 3 clusters. And when we plot the PC1 score value in space, we recapitulated the pendo samples. And so we were able to confirm this data from like the previously published paper that defined the boundaries of the pendoclone with microsatellite data. So next, we wanted to identify the somatic mutations. So as I mentioned earlier, usually we focus on germline mutations, which are the mutations that would have been present in the seed and that are just shared by every ramet in the clone. So the only mutations that will tell us about the evolutionary history of the organisms are the somatic mutations, the one that emerged after germination. However, we do not have access to these mother seeds, the control. So how can we label mutations as somatic? So to do this, we came up with a set of filters. The first set of filters to delete the ancestral mutations, so the ones that would be shared between every ramet within the clone. So we removed the common mutations that we found in pendo and then the neighboring clones. And we also removed mutations that were present in more than 80% of the samples. And then to get rid of some of the technical artifacts, we removed variants that were found in a single sample. And because we're dealing with somatic, which are rare mutations, we were expecting a high rate of false positive or negatives. So we need to assess our ability to recover these rare mutations. And to do this, we used this replicate data set where we sequenced 8 times the same DNA extraction. So I applied the filters that I presented to you earlier, and I found a candidate somatic mutation set for these 12 samples of 101 mutations. And what we found is that if a mutation is found in 2 out of the 8 samples within the group, it was found in approximately 44% of the rest of the replicate. So this number is pretty low, but it was still better than a random signal. This is the distribution from a randomized data set. So despite being rare, these somatic mutations replicate between samples. Now that we know that we can trust the data that we're working with, I looked at the spatial structure within the clone. Because And because the tree expands by the root, you would expect trees that are closer in space to also be closely genetically related. So we looked at the relationship between the genetic distance and the spatial distance. So here you can see the number of mutations that are shared between pairs of trees as a function of the spatial distance between these same two trees. And we find a slightly negative correlation, which means that trees that are closer in space are also more closely genetically related, and this was significantly different than expected by chance. So this is from the point of view of the ramets, so every mutation that is shared by two ramets. And then we took another approach, which is the approach of the mutation, looking at the mean distance within groups that share one mutation. So I took like every mutation and I look which trees have this mutation and what's the mean distance between these trees. So I obtained this distribution, and when compared to a randomized data set, we saw that it was also lower than expected by chance. So mutations cluster in space, a little less so that we were expecting, and I'm happy to chat more about this after the talk. So finally, we set out to estimate the age of the pendoclone, and we did this using BEAST, which is a program inferring phylogenies based on sequence data. And we did two estimates for now based on two different assumptions. The first assumptions being that currently we detect all of the somatic mutation. So I converted the phylogeny height to an age, and this estimation tells us that the pendoclone would be about 34,000 years old. However, and as showed you earlier, we do not detect all of the somatic mutations. When we compare the mutations that we found in the replicate dataset to the dataset they came from, we only find 6 mutations in common. So we know that we're missing about 94% of the mutations. So the next simulation that I did was taking into account the fact that we're missing most of the mutations. And to be able to take into account these missing mutations, we first need to know the relationship between the phylogeny height and the proportion of missing mutations. So I took the whole data set, and then I removed a certain proportion of mutation from the data set to find this relationship between the phylogeny height and the proportion of SNP that we would be missing. And I used this relationship to come up with this second estimate, which would age the pendoclone of around 80,000 years. And of course, should we believe these numbers, because it is a pretty old age. And so looking at the literature, we know that clonal organisms can reach ages that are about the same time scale. And I think what's interesting too is that there is some core data that's been done on the lake that's just right by the pendoclone. So this is pollen data. And this green box here is Populus, which is the genus. And we see that there's evidence of Populus presence in the last 1,600 years in the Fish Lake National Forest. So there's some evidence that, at least in the last 1,600 years, there was the presence of Populus in the area. So to conclude, the pendoclone may be at least 30,000 years old. There is a weak genetic structure which suggests a lower spread of somatic mutations than we expected. And next, and I'm currently still working on this, I'm interested in comparing the somatic mutation structure and rates between tissues within ramets. With this, I would like to thank the people that helped me get there, and I will take any questions.